And this allows you access to FAANGMine itself, the data mining warehouse, but then we also have genome browsers for all the different species that we have in FAANGMine. And there's a documentation, examples, and then an announcement page. So I'll be showing you a little bit about JBrowse later today. So to enter FAANGMine, you just click here and you get to the actual FAANGMine interface. So the first thing you do if you're new to FAANGMine is to create an account. And the reason why you would do that is because you can save lists of identifiers and that result from your analyses, or you can upload lists of IDs and be able to save those to be able to use in queries. And so the way you create an account is by clicking login, and then you'll get to this page where you can say create account now, which I'm not going to do because I already have an account, so I'm going to log in. There. And so then when you're logged in, it will have your username up here. So this is my FAANGMiner is my username that I use for creating tutorials, but you should actually use your email as your username, and that allows us to for you to get announcements when we're going to put out a new release. So, oops, sorry. So this is the homepage. At the top of the homepage is this navigation menu that has these different tabs for the different kinds of tools that FAANGMine has. And there's a place where you can just do a text-based search of any kind of keyword or identifier, and it will bring up a report page, or you can have a list of IDs that you can search for. And then in the middle, we have this bar going across, and this we call the template bar. It has template queries. So these are different queries based on just different kind of categories of information. And they include just menus where you can enter information to do queries. And I'll go back and show you those later. The first thing I actually want to show you is the data sources tab, which is, it provides all the data that we have in FAANGMine, including the publications and FTP links for the sources. So basically, we have genome assemblies, and then we have genes from RefSeq and Ensembl, homology from a couple different sources, proteins from UniPro, protein domains from InterPro, gene ontology, which comes from UniPro, and pathway information from KEGG and Reactome. We have gene disease associations for OMIM, and so you can actually use orthology to then, compare to identify these diseases that might be pertaining to your ortholog in your species. Interactions from BioGrid and Intact, QTL from Animal QTL-DB, variants from either Ensembl variation or the European Variation Archive. Variant effects on genes from Ensembl for the species that are in Ensembl. And then we have, for the SNPs, we have alias IDs that come from SNP chips. And then what's new in this is that we have a lot more FAANG data. So we've always had RNA-seq-based gene expression levels, but we've added some new data in this release for RNA-seq. And but what's the newest thing is a lot of this FAANG data. So in the previous release, we had histone marks for horse, but we didn't have other FAANG data yet. And so now we have a lot more for other species in addition to horse. And so we have chromatin accessibility, which is from ATAC-seq or DNase I hypersensitivity. We have some chromatin architecture. So this are the AB components and TADs, chromatin states, CTCF binding sites, histone modification marks. And so the sources are all here, or the publications are here, and then the actual sources of where the data came from is actually, there's a link over on the very last column. And then we also have PubMed publications, and there's a lot of different ontologies. So I'm now going to go back to the home page, and I'm going to, at the beginning, I'm going to start off focusing on this FAANG data for in case people here are more familiar with FAANG, so I'm going to kind of focus on the new stuff. And then later I'll go into kind of more things that I've taught before. And so if you click the FAANG tab, there's a lot of information about the FAANG data itself and then how we model it in our database. And so when we create this database, we need to use terms that are in the sequence ontology. And so that kind of constrains us a little bit. For example, there's not really a term in sequence ontology for AB compartment. So we've had to use the general term biological region. And then for the different chromatin states, regulatory region. And then histone modifications had to be called histone binding site. So this just explains all that. You don't, I guess, you probably don't need to memorize all this in order to be able to do this, but this is just kind of for background for you to understand, and especially for doing the region search to know which features to select. So let's see, I'm going to click on organism to all FAANG experiments. And so this opens up a template query and where you can select a different organism. I'll leave it on this one. And then it's automatically set to show FAANG things that come from FAANG, but we also have legacy data, which would only be RNA-seq data. So we'll just leave it as FAANG. And before I click show results, I'm gonna click on edit query, which brings you to the query builder. And so this is actually the tool that we use to create queries. And it can be pretty complex, but I wanted to show this to you because it kind of can, it gives you sort of an overview of all the different kinds of metadata that we have to go with the FAANG data. And so in this query is actually searching for experiments. So we have experiment is rooting this tree over here on the left. And then so you can see all the different kinds of information we get. So we kind of take a merge of what's available in that we get from EBI, and then for some of the legacy data, what we get from Ensemble. And so we get experiment information. And so the experiment information comes mostly from the SRA at NCBI. And then we have information about analyses. And so this actually comes from FAANG. Then, just bioproject information. And then biosample information. And so this is sort of a merge of NCBI biosamples and EBI biosamples. But whenever possible, if it's available in EBI, we prefer to use that because that is data that was submitted mostly through the FAANG portal and therefore has really good information such as ontology terms. And so we have all these different things. And so everything over here on the left side that's in this little light blue square or rectangle is actually a column selected for output. And so these are all the columns that would be output based on the way this query was built. And so just as an example of just a simple making a change to the query, if I wanted to add the biosample submission title, I could just click show here next to submission title and it gets added. And so then you can see that at the bottom here on the right in the query overview. And then if we scroll down, we can see all the columns or fields that were selected for output. And these can be reorganized. For example, if I want this submission title to be over here right after the bioproject ID, I could do that. And there it's moved. And so if I click on show results, then I get to this tabular output, which actually kind of goes over. You can scroll back and forth if it's too big for the page. And here all of these columns have all these little icons on top that allow you to do different kinds of things. For example, you can sort a column. Let's see, let's say if I sort on biosample name, I can sort it and then it changes. Or I could undo whatever I just did with the undo button that shows up every time you do something. I know that there's actually, for the pig data that we have in FAANG, there was no information about the sex of the biosample for the biosample. So I can just remove that column with this X. There's also, I could hide a column with these little dots. So like if I click this, then that column sort of disappears. There's a histogram that actually provides information about what's in the column. So, so this is the information about CHIP antibodies. So for a whole bunch of them are an RNA-seq, so there's nothing there, but then these are the things that actually have CHIP antibodies. And let's see, and the one thing, the last thing I want to point out here is these two columns, the biosample ID and the specimen tag. And so they look the same for some of the rows, but they're different for others. So what a specimen tag is it ties all the samples together that come from the same individual, the same kind of tissue of the same individual animal, but done with different kinds of technologies. And so that allows you to pull information together for, you know, if you wanted to, like, for example, look at all the different histone marks along with the RNA-seq and ATAC-seq or something like that. And so depending on how these samples were submitted to the biosample database, sometimes the biosamples were submitted as the tissue sample, and sometimes they are submitted as more related to the library, or and so there'd be different biosamples submitted for different kinds of experiments. And so if we look at the column summary for the specimen tag, and these always show up in alphabetical order, so you can see that some of them are not biosample IDs. And if we just click one of them and to filter for it, there. So these are, so now we can see all of the information for this one individual adipose in this animal that was rep P38. And so that way you can actually get this information together. Okay, let's see. So continuing on with, oops, sorry, I keep doing that. Continuing on with the FAANG data, we're going to go and do a region search looking at FAANG data. So the regions search page was modified, I think, probably in the last release, where you can select different kinds of types of experiments that you might want to get features for in your region search. And so we're going to do this for pig. And if I just click on this square here, it actually automatically selects all the different experiments available for this thing, for like CTCF binding. All the experiments are selected. You can, you can undo that and select one at a time. But for now, I'm going to just select all of them so that I can show them to you. And so we'll do all the chromatin accessibility, their chromatin architecture, all the chromatin states. And so chromatin states, they come from two different publications. So you'll see there is one publication where they predicted 14 chromatin states and another one where they predicted 15. So they're labeled like this. And then histone marks. So you need to, whatever kinds of experiments you want to select, you need to make those choices before you come down all the way down here and select what feature types you want. So the feature types showing up here are dependent on what kind of experiments you select above. So if I select features here and then I go up above and select different things afterwards, this is whatever I selected is going to be erased. So you have to do this in order. I'm going to select all the feature types that are related to this kind of FAANG data. And so the biological region, remember, that's related to the TADs, the AB compartments, and then the CTCF binding site. And then we have open chromatin region. So that would be ATAC-seq. And then for chicken, we also have DNase I hypersensitivity. Let's see. So we have histone. And then the regulatory region is what we would select if we want to see the chromatin states. And I think that's, oh, here, topologically associated domains. And so these are all these features are the types corresponding to the FAANG data. And so now I'm going to, let's see, I'm going to just paste in a few SNP positions that were related to a GWAS study or some GWAS results. And so these are, there's a few different formats you can use for a region search. I like to paste it in from an Excel spreadsheet, so that's tab delimited. You can also do this one where there's a chromosome and a colon, and then this to the start, dots, and the end, and so on. So these are the other formats that are allowed. And notice that even for SNPs, you have to have both a start and an end position. So those would be the same position, the same coordinate, but they both have to be there because the way the region search is set up is you have to have a start and an end no matter what. You can extend your region on both sides if desired, but I don't want to do that, so I'm not going to. And then you also can do your search in a strand specific way, which you could by checking that box, but I'm not going to do that. So I can click search. And then, so this actually ran pretty fast. Well, first of all, because it was a small search, and also because I did the search earlier, and so these searches are cached on the server for a while. So some of your searches, if you have a lot of regions or very large regions, it might take a while. But then once you've done them, they'll be cached. And so then if you do them in within the next few days, you can redo them and they'll be fast. So this is showing the results that we have. So there's in this output table, it's divided up into different sections according to each region that we entered. So we entered 4 regions, which actually 4 SNPs, so each one will have its own section of the table. So here, this one long section is for the first SNP, and you could download different kinds of files related to this one region if you want to, or you can create a list, and we'll be going over the list tool later, but you can create a list of IDs. But just to look at these, so this output shows you both the type of how we named this data in the database. So every single feature that was identified that's in a bed file, bed or whatever kind of format file you have for the FAANG data, every individual element was given in both a unique identifier and a name. And so, for example, the chromatin states were given unique identifiers for each one, and then just the name, the name of it is a state. And I know that quiescent and low signals are not actually regulatory regions. They're just, I guess you could say non-regulatory regions, but these are still in here because they might still be of interest. For example, if you especially want to look at what's tissues where you have low or no signal, and then other tissues where you do have something. Let's see, and so here you can see some histone marks that have, so that we named them with these kind of long names that give a lot of information, the chip antibody, the tissue, there's an individual name, and then there's actually the peak ID that came from the bed file. And then, but then there's also another unique identifier that includes the experiment ID and just a numerical identifier to make sure it's unique. And so there's all the results for these 4 different SNPs that we entered. And so this is really a lot of data. So normally you might not want to do all of these, search all these data sets at one time, or you might want to search all of them and look for something interesting and then repeat the search more narrowed down. But one thing that might be useful is that you can, if you go to the top, you can download the results for all of the regions together. And so, for example, you can do a tab delimited file. And let's see. there. And so here's, you know, this is the file that I just downloaded that has all this information, including the actual input that I gave to do the region search. And so these, since I did a region search of coordinates, I actually don't know the IDs of these SNPs. But if I wanted to know the IDs, which I actually did before, is that you can do a region search with the these coordinates and then just select SNV as the feature that you want to search for, and then you'll find these SNPs in the database. You'll find out their IDs, and you'll be able to save a list of them if you wanted to do that. So another thing you can do on this page is you can actually save a list of these of different features. I didn't really think that was that useful at this point, and so I'm not going to show you how to do it right now. And another a little bit of a disadvantage is that you can only save one thing at a time. So we're going to go back and do a region search later where we're actually going to save a list and do things with it. We're not going to save these. But what I do want to do is show you viewing this data in JBrowse. So I wanted to go down for this this SNP, which overlaps an active enhancer in liver. So I want to look at that. And so what I'm going to do is, I'm going to just copy paste this this actual SNP location, and I'm going to go to JBrowse. And so if I just enter that location here, so you could just ignore this chromosome ID here. You can just enter the location and click go, and it goes straight to that in JBrowse. And so of course it's, you know, the browser can't go to one single coordinate because, you know, that's impossible to do. The browser is too wide to do that. But that SNP is actually in the very middle. And so if we go to select tracks, and then this is a faceted track selector where the data types, there's categories on the left side that can be used to filter the data. And it's divided up into either a data type or organ system or bio project. And so we're going to just click down here, variation. And so we can find the SNP track. So we can select that. Then if we go back to the browser, the SNP in the middle is actually the SNP, the actual SNP that we searched, which I verified by looking for those IDs separately. And so I'm going to, by clicking on it, it just makes it highlighted in red. And I want to do that because then when I zoom out, I'll be able to still see it. And now I'm going to go and select some other tracks. So I want to select histone marks. So I'm going to just highlight all of these, and then also chromatin state. And then, so I'm going to search for liver up here and under contains text. And so here, these are all the FAANG samples for that data. And so there were 2 individuals that were done. And so there's a rep P348 and rep P350. And so we'll just select both of those. And then we can just click up here on top of data next to data type, and then select all these tracks and then go back to the browser, and there they are. So we, so we'll like kind of slide up so we can still see our SNP, and then we'll start zooming out so that we can kind of see other things. And so, but you're probably more interested in seeing what genes are around, so we can go back and let's we'll click this red X to clear out this contains text, and then we'll unhighlight all these things, and then we'll select gene prediction. And so we're going to select ensemble protein coding and RefSeq protein coding, and then we'll go back. And so now we can see these genes at the bottom. And so there's a lot of stuff here. So I'm gonna remove some tracks. For example, there's none of this H3K27ME3 right here. So I'm gonna just get rid of these. I can X these out. And I'll just leave the other ones here. So you can go down and see everything that this SNP is overlapping. And there's the enhancer. And then you can see this, this nearby gene. And then another thing we can do is if we, let's see, let's undo gene prediction and variation. If I enter liver, and then I'm gonna, oops, not that one. If I, just to show you, I'm picking this, one of these histone mark tracks to show you that there's a specimen tag here. This is the same specimen tag that I showed you in FAANGMine. And so this is the, these are the tags that allow you to find other tracks that are related to it. So you'll see that all the tracks are these two specimen tags. But what I want is this, the RNA-seq. So I want to get some RNA-seq XY plots. And if I want to get those specific samples, then if I just start typing in the specimen tag here, then here I pick up both of them. So I can just select these. So these are the RNA-seq that go with these samples. And so now you can view expression also along with this other data. And so this is basically a way of exploring your data in the context of FAANG data. And I think that it's mostly probably useful for, in this case of where you just want to investigate some of your GWAS SNPs or something like that. FAANGMine is not really meant for a large scale integration of data where you wanted to, you want to like get the whole genome data sets for everything and then predict chromatin states or something like that. So that's, it'd be best, to do that for you to get just the full files, input files, rather than the doing this kind of a region search in FAANGMine. Okay, so does anybody have any questions related to that so far? Okay, so now I'm going to go on, and I'm actually going to go back to some of the tools that I've shown before for the people who are new to FAANGMine. So we're going to go back to the home page, and what I want to do first is to show you the list tool. I keep doing that, sorry. This is a new laptop and I'm not used to it yet. Okay, so I'm going to click lists. And this brings you to a page that it will either bring you to this upload page where you can create a new list, or sometimes it brings you to the view page where you view your existing list. But to go back and forth is this upload and view over here in the upper left. So I'm going to go to upload. And so for this, I'm going to use, I have differential expression data from a published data set. And this data set provided both, it's from PNAS. It has both it has all genes that were expressed in the experiment, and then it shows their expression levels. So we can, if we enter a list of all the genes that are expressed, we can create a list and use that as a background gene list for gene ontology enrichment. So we'll paste these in, and this is bovine. So we're going to select bovine just to show you this select type. There's a lot of different, anything that has an ID, there can be a list uploaded for it. So there's a lot of possibilities, but gene is probably the most common one. Then we'll click create list. And so this was a really big list. It was like 10,000 genes. And the reason why this went so fast is that I ran it yesterday. And so it will, the first thing you should do is probably give it a name that makes sense to you, because otherwise it gives you this default name. And so I'll call it bovine background list. And you'll see that it says I entered 10,833, but it only founded 10,285. And then at the bottom, it lists all the ones for IDs not found. The reason why these IDs were not found is that this is an old paper And I think it was done with the UMD 3.1. And so a lot of the ensemble IDs, got lost in between assemblies, but there's still a lot of IDs there. So we're going to save this list after we gave it a name. And so now once I did that, it's actually saved now for me. And then, so this is going to bring me to a page that has a table that just gives information about the gene list that I entered. So this is just default information. And if you scroll down, you'll see that there's going to be an automatic gene ontology enrichment. And so this enrichment, or it's gene ontology, publication, and pathway enrichment, this is done automatically whenever you enter a gene list, but usually you don't want, usually you want to just ignore it until you make sure, you need to make sure that your list only has genes from only one gene set. So if we see, look here, there's actually, most of the genes are ensemble genes, but there are some that are ref-seq. And so in order for the gene ontology enrichment to be statistically valid, you can only do one gene set at a time. And so we're going to pick ensemble since that's most of them. And then we can choose that, and then we can restrict table to matching rows. And so now we only have ensemble genes. And so we're going to save this list. There. So we gave it a new name. And so this is a new list that's saved. And so that's going to be our background gene list for enrichment. And so we still don't even want to look at that enrichment widget because this is not what we want to analyze. So we're going to go back to the upload page, and then we'll get these are the differentially expressed genes, which I've already prepared the list to make it fast. And so we'll create this list. This one's going a little bit slower. In the meantime, does anybody have any questions? No. Okay. Okay, I think I'm going to just be impatient, and I've already actually saved these genes when I was preparing for this tutorial. So, oh, okay, wait a minute, here it is. We'll just go on. Okay. Okay, again, we lost some genes, but that's okay. So we'll save this list. And again, we have to make sure that we're only doing one gene set. So we'll select ensemble, restrict to matching rows. And so you'll notice that there's, after we do say restrict to matching rows, or we've only have ensemble, it does the gene ontology, but we can't, this enrichment is based on the original list. So when you filter the table, it doesn't actually recompute the enrichment. So what you have to do is you save a new list. And then when you click on the name of the list, like we can just click on it here. We could either click on it here, or if we wanted to go to our list of lists and view them, you can see that it's here. So anytime you click on a list, that's what activates computing a new enrichment. So then when we click on it, and now the enrichment is recomputed. And so now we can go down to the widgets, and so we can change our background population to the background genes. And so ensemble doesn't have any KEGG pathways. All KEGG pathways are attached to ref-seq genes, but ensemble has reactome pathways. So we change the pathways to reactome, and then here are our results for the pathway enrichment. And so you can change your test correction or your p-value. You probably wouldn't really want to make your p-value any larger. And then this can be downloaded as a tab delimited file. And then here again, so your background population needs to be adjusted for every single enrichment widget separately. So even though we did it in pathway, we still have to do it again for, let's see, for the gene ontology background genes. Let's see, where's the one? Well, this is the one I did yesterday. That will work. So the sometimes the background doesn't make a lot of difference, and sometimes it can make a big difference. It especially makes a difference on the marginal, on any of the marginal things. So it is really important to like change the background. Sometimes you don't have a background gene list, like you know, like especially if you wanted to redo a published something that was published, and they don't actually provide all the genes that were expressed in the experiment. And then what you would do in that case is you would actually just choose the correct gene set. So ensemble genes, all the ensemble genes, so that you're not including ref-seq genes in your background. Okay, so that was we did our analysis using ensemble genes. If we wanted to, we could also see what happens with KEGG pathways, but we need ref-seq genes. So to do that, we can get cross-references for those genes. So if we click on alias and dbxref, there's a template called gene to cross-references. And then, and so what you can do when you have a list is you, rather than entering a single ID, you can click constrain to B, and then normally you'd want to leave it as in. And then you can choose from the pull down menu, your list. And so all of the species have their entire gene set as default, you know, available by default, but then also you'll see the list that you just made. And so let's see, background. I can't find the one I just made, but oh, wait a minute. It's down here, bovine. I don't know. This, sorry, I shouldn't have so many things in my list. I have too many choices. I'm going to just choose this one. This is the one I made yesterday, the background gene list. It's the same, so I'll just choose that one. So I'm going to create the database cross references for that, the background, and I'll just click show results. And so so this shows all the ref-seq. These are all ref-seq genes that go with these ensemble IDs. And so I can say save as list, and then when I do that, I need to make sure that I'm highlighting this part where it's the cross-reference, not the actual gene. So I click this line here. If I click on that, then it will allow me to save those. So and then I would do the same thing with the differentially expressed genes. Yeah, these are in alphabetical order. So the ones I did yesterday that are labeled with B Taurus are showing up first. So I'll just select these to make it go faster. Oops, it's the one for ensemble I want to select. So we'll save these. So you can see that there's that. I can actually just click on this list from here to do an enrichment. So this time we actually do have KEGG pathways, and then we want to change this to be the appropriate. Let's see, where's the? There it is, bovine background ref-seq. Okay, so that's a pretty common thing to do in FAANGMine. Let's see. So now I'd like to show you how to do similar kinds of like saving lists when you do a region search. So we're going to go back to the region search. This time we're not going to search for any any of the FAANG data. We're going to just search for gene. So the features that show up by default in these where you can select a feature, those are all things that are related to gene annotations on the assembly that come from Ensembl and NCBI. And so none of the FAANG features are actually showing up in this list right now. And so let me get my data set. So these are QTL for bovine respiratory disease susceptibility. And actually, I want to search for gene, so I click gene. I paste in my coordinates, and so these are actually classical GWAS. So I'm not going to extend the regions. I just want to find genes that are within these regions. So you can see that it takes a while with longer regions. It's still going, but you can partially see the results. And so you can see that there's each region shown separately, and then the genes, and you'll see that there are both Ensembl genes and RefSeq genes showing up in this region search. So we'll wait till it's totally finished before we can save a list. So now, see, now that it's finished, you could either download it, tab delimited file, or you can save a list. So this create list by feature type, it only shows one feature because we only search for one. And so we can just click go, and it saves the list. And so in this case, it doesn't actually allow you to give it a name. Instead, it's going to just give you a default name. Pretty much all the genes are going to, whenever you do a regions search for genes, it's going to always say all regions gene list, and then there'll be a number. Okay, so here we have both RefSeq and Ensembl genes, about half and half. And so you can see this gene ontology enrichment is done, but we need to ignore that. And that's really important. And a lot of times you'll see gene ontology enrichment when you have both of your gene lists there. But then when you analyze the list separately, RefSeq separate from Ensembl, you won't have any enrichment. So it's really important that you actually do that, because otherwise you're going to have false results. And so I want to just scroll down below this table. There's actually information here that you could read. You should read always to remember to do that. And that reminds you to separate your gene sets. And so here we'll do similar to what we did before, where we're going to first we'll filter for one gene set. We'll save the list. So we save the RefSeq one, and then so we can do use this undo little arrow here to undo the filtering, and then the Ensembl genes are back again, and so then we can click, and there's and this time select Ensembl and restrict table to those rows, and then save those genes. And so now each one of these, you can do the you can do your enrichments by clicking on these, either from here or by going to your list view. So if we click on RefSeq, so clicking on that now caused the analysis to be redone. And so you'll have more correct results, but you still need to change your background population, because by default, the background population is it's the gene ontology annotations for all the genes in the entire database, which is a combination of Ensembl and RefSeq genes. So you need to still change that. And in this case, since we're just doing a region search, we're going to just use the whole genome as the background. So we'll select Bos Taurus RefSeq all genes, which is available by default in there. And so you can see, like, if you watched, it doesn't look that much different, but actually the p-values are different. And it's possible that the very, the most marginal things could actually disappear or change. And so then you'd also have to do this separately for your ontology. Yeah, I think that one did change. I think some were lost at the bottom. And then also I forgot to point out before that you have your separate gene ontology divisions. Okay, so I'm going to just show you a few other little things. One thing I want to show you is the importance of knowing which chromosome IDs to use when you do a region search. And so this is always a pain for everybody in our community, is that there's different identifiers. And so I'm going to show you how you can match your identifiers to what we use in FAANGMine. So there's under entire gene set, there's a template called Genome Assembly to Chromosome IDs and Lengths. And so if you click on that, you can just select a genome, show results, and it shows the chromosome IDs that we use. So it reminds you up here, actually it doesn't tell you. So these are, anyway, the chromosome IDs that we use are the ones that call chromosome ID. And so we use, we get this information from the assembly report at NCBI. And so NCBI has a first column. The first column is always the chromosome name that was submitted. And so this comes from the sequencing center. And then there'll be a column called assembled molecule. And so that's where we get the, that's where our primary chromosome ID comes from. Oops. And then we have some when we don't have assembled molecules, but we just have unassigned scaffolds, those use RefSeq IDs. And the reason for doing that is because it matches the RefSeq GFF that we use. But you can see also, these are all the other possible things. So you might have these IDs. You might have, these are GenBank IDs. And so, and then over here is all of the RefSeq IDs. So even the RefSeq IDs for the chromosomes. So you can download this. If you click export, you can download this as a tab delimited file, which you can open in Excel, and then you can use this to change your IDs if you have a spreadsheet of information. Okay, so the last thing I want to, well, actually I want to do, I want to show you just a simple report page. So I'm going to just go to my list view. I'm going to click on one of my lists just so that I can get a gene ID. So if I just click on an ID here, I can go to a report page. And then, and this is a similar kind of a report page that you would get to if you use this ID or the gene name or something, gene symbol, or like a description in the keyword search. And so I just wanted to show this to the new people who I haven't shown before, is that this has pretty much the kinds of information that you'd find in other databases when you have a gene report. The main difference is that everything is in these kind of tables that can be downloaded. And then some of them are not opened up by default, and so you have to click on them like this. So this one only has one transcript. And so, but it's still a table that's closed by default. And then there's the transcript ID to the protein ID. Let's see. And so anytime you see one of these little green things here, that means you need to click on it to see the actual table. And then there's some other things where the tables open by default. So I just wanted to show you that we have this report page with different information. And then the last thing I want to show you is how to build a query from scratch. So we're going to build a simple query. So for that, we're going to go to the query builder. And so when you first get to the query builder, there's these different sections. So one of them is where you can browse the data model, which actually just kind of, I don't know, I think it's actually confusing, but it does give you an idea of all the different kinds of data that we have and how many data points of each one we have in the database. But that's not the best way to start building a query. So I'm going to go back. let me show you another thing is that you can import a query, which I'll show you how to export one in case you want to import one later, but you do that there over here under import query. And then before I start actually building a query, I'll also show you that you have a query history for all the queries that you did in kind of like in the recently. So it's queries during this session, but I think I have some that were like not to like, I don't know how long this is saved, like maybe a couple of days. So this isn't saved permanently. If I wanted to save any of my queries permanently, I could click save and then it will be saved in my account. Otherwise, it will be saved for a few days. But I'm going to start building a query over here where it says select a data type to begin a query. And so there's a lot of ways, different places you can start. And so the one that we saw before had been started with experiment. I'm going to start this one with gene, which is pretty common. Select. And I want to constrain the gene IDs. And you could either enter an ID here or you can click contained in. And so we can select a list. So we'll just do what are differentially expressed genes. Let's see. I'll choose that one. Can add that to the query. Click add. So you can see now they're going to be constrained. I also I actually want to see my gene IDs. And so I need to click show over here to say show. And so now this is saying that I'm going to constrain my IDs to this list and I'm going to show gene ID in my output. And so for this, I want to get pathways. So I can go scroll down and click on pathways. And I want to get the ID and the name. And I'm not going to select the data set because I already know for all the RefSeq, it's going to be from the KEGG data set. So I won't do that. I'll just do this for now. And then so there we have a simple query. And then, I could rearrange these things if I want to, like this. You can also click on these arrows if you wanted to sort in some different way. Click show results. And then there's your query. So, so if you noticed when we say save as list, we only have 1,500 genes, but I remember that there was over 2,000 genes that were differentially expressed. I think maybe 2,900 or something like that. And so the reason that they're not all there is that not all of the genes have pathways. So if we want, I have a chat. Oh, okay. Bye, Krista. So we're going to make some changes in this output. So we're going to two things. First of all, I forgot to add the gene symbols and so I'd rather I want to look at the gene symbols because that helps me know what the genes are. So I can click on manage columns and then click add a column and then here's symbol right here. Actually, I want to add description also in case there's descriptions. So I can click add two new columns. And so you can see here that they've been added. Actually, I want my symbol and my description to be up there close to the ID there. So I've rearranged the order. Now I can click apply changes and they're here. So now I have my gene descriptions and my gene symbols, but I still don't have all the genes there. I still only have 1,500. And so what I can do is I can click manage relationships and say that I want the pathways to be optional. And then that way everything will show up. So, and so now all of the format of the table has changed. So all the genes are here. If we click save as list, we can see, it's 2,700 genes. And then the format has changed so that under pathways, whatever is optional turns into sort of an embedded table. So if you click on it, you can see it and you can close it. And then some of them have 0 pathways, but they're all there. This little thing, sometimes they show up and I can't get them to go away. Anyways, if you want to export this into a tab delimited file, this will turn into a normal format where the genes that don't have any pathways, they'll just have blank cells. Let's say I also wanted to add more information. I can either add more information using manage columns, or if I click on query, I can actually go back to the query builder. And so notice that this has been changed because you can see the gene ID and symbol are added. Then you can also see next to pathway collection, if I click on this little symbol here, it shows now that it's been constrained, it's been set so that all genes show let's see, show all genes and show pathways if they are present. Basically, it shows all the genes no matter what, whether they have a pathway or not. And so that can be changed here, or it could be changed where I showed you before. Let's say I want to add Go annotation. I can open up Go annotation. I can I want the ontology term. I want the ID, the name space to know which ontology it's from, and I also want this to be optional so that some of the genes that don't have gene ontology won't disappear. So I can click on this here where it says inner join, but I can click on it and I can change it to say show all genes and then add to query. So then that's an alternative to using manage relationships. Oops. And sorry, I keep doing that. And before I click show results, I wanted to show you one last thing, is that if you click on export XML, this is the XML that you get that you can use for importing a query. So if you wanted to email a query to somebody, you can copy paste this into an email, and then they can use it and be able to import it. And there's also here where you can get code for the API if you wanted to do this programmatically. So this actually creates code for this specific query that you can include in your scripts. So if I click show results, then you'll see that it has been changed, and so now we have all the Go annotations. So that is all that I wanted to show you, and